Okay. In the last class, we have mentioned about fermentation of yeast, and while preparing the yeast culture, first you need to prepare seed culture in multiple reactors, and then finally you transfer the seed culture to the main fermenter. And generally in main fermenter, when you try to get the yeast, you try to get a concentration of say 6 million cells per ml. So to get that much of yeast culture, you do multi-stage seed preparation process, and you follow multi-stage seed preparation process, and finally you add that to the final fermenter. And in the fermenter also we have mentioned mostly we use molasses, either cane molasses or beetroot molasses, and also you add some other additives like ammonia or uh, some phosphate solution to provide sufficient amount of nitrogen and phosphate. And finally, when you get the broth, means if you operate the system in a fed batch mode for say 20 hours, then you will end up by getting a concentration of 5%, say 4.5 to 5% biomass concentration. The solid content is around 4.5 to 5%. Now, this solid content is the maximum achievable solid content in the fermenter. So after getting the solid, you centrifuge it to just remove the residual liquid, and you just concentrate the solid. And after centrifugation, whatever pellets you get, that you just wash with water, or you can say resuspended the solid solution, solid material. And while resuspending them in the liquid, essentially you do two things. One is washing the uh, solid biomass to get rid of all residual broth, and also you try to reduce the temperature. So essentially this entire operation will be done in a cooler, and then finally you get yeast slurry, and that's called yeast cream. So yeast slurry or yeast cream will actually have uh, yeast solution, and when you get this yeast slurry, this can be used directly in other industry, like if you need to use in brewery industry or winemaking industry, there you can directly use the yeast cream, where you have yeast suspended in liquid media, or when you try to preserve them, so whether it is at room temperature or at 4 degrees centigrade, there you need to further process the yeast cream to get different kind of final product. So one option is to remove the moisture to certain extent, which will allow you to grow the yeast uh, in subsequent process very quickly. And when you try to remove this yeast, it removes the moisture. You just use plate and frame filter to get the cake, and then you use mixer to just have uniform size of the broken cake, because after dewatering, in the last class we have mentioned about plate and frame filter. So in the plate and frame filter, you will actually get solidified cake, and when you remove the cake from the gap between plate and frame, you will actually get broken biomasses. So now you put them in a mixer to get almost uniform size, and then this biomass is actually pushed through a extruder. So when you push this biomass, because the biomass also contains a lot of moisture, so you push through the extruder, and then you get different shapes. So you can get a solid rectangular shape. Type of extruder you use, that will actually guide you to get a different shape. So if you get a cylindrical shape, then you can just pack them in as a cylindrical rod form. Only thing is moisture content is significant, and there is a possibility of contamination by bacteria. So to avoid that contamination, sometimes we use some solution like before processing it in the dewatering, before dewatering in the same stream, we can add some phosphoric acid solution or ammonium persulfate to make sure it will not get contaminated by other bacteria. So ultimately, you are just altering the pH in such a way that it will not allow any other organism to grow. And then you pack them pack them in a either plastic bag or paper packets, and then store them at 0 degree centigrade to zero, in between 0 to 4 degree centigrade. Now this compressed is very easy to grow or propagate in the second process. So after getting the compressed yeast, you can just again directly add it to the fermenter, and then within very short period of time, this yeast will get activated and it will start fermenting the broth present in the other reactor. So advantage of the compressed yeast is that you are not putting too much of effort to remove the water. So moisture content is high. So the yeast will be active, and you can readily use them in subsequent processes where you try to use yeast as the inoculum to do certain bioprocess. Now, active dry yeast is again another mode of getting means biomass, a live cell, where you do little bit more dewatering. So essentially, you try to keep 8% moisture. So in the earlier case, for compressed yeast, you still have 15 to 16% moisture. So compressed yeast, preferably it is better to have around 10 to 15% moisture. There you are getting around 8% moisture. So active dry yeast, there also you dry them. Only thing is the dewatering process is actually quite similar. You can just dry them in or remove the moisture using plate and film filter or centrifuge, and then you put it through the extruder. So here the dewatering process moisture content is significantly high, say around 15 to 20% after dewatering. Now when you get a dough kind of material, you can just easily put it to the extruder and it will make very long strand. So you have a moist dough kind of material. Now you put it in a extruder. Extruder, suppose you have in the extruder device, you have a specific pattern. Now if you using a compactor, if you just press the dough through the extruder, you will actually get long strands. So the kind of pattern you are using in that extruder, that will actually guide you to get either cylindrical or cuboidal or different kind of strand. So these fine strands are actually kept on a plate or tray, and then using conveyor belt you move this tray to multiple drum dryer. So you have one dryer unit, and then you have another dryer unit. In this way you can have 3-4 dryer units in series, and then this plate will actually the plate will be placed on a conveyor belt, and conveyor belt will take the plate from one dryer to another dryer. And in this dryer, you try to maintain hot air flow and the temperature of the hot air is maintained around 25 to 45 degrees centigrade. Hot air to remove the moisture. So obviously, when it get exposed to this warm air, then it will remove the moisture from there, and in during this process, it will also get the color of the yeast will also get changed a little bit. But as still this moisture content in this yeast material is about 8%, so still you can get live cell present in this. Only thing is the shelf life of this particular material will increase because moisture content is less. And here also you get substantial amount of nitrogen like the other cases also, and you can directly use it mostly for baking. So when you get this dry yeast material, then you can just use it as a ingredient for baking. And again, when it will get sufficient amount of moisture and also carbon source, it will start working on the carbon, and then it will ferment the carbon and produce carbon dioxide, and that is the way you actually get the fluffy bread. So first you prepare the dough, and while preparing the dough for the bread, you just add certain amount of yeast, and sugar would be always there. So you just and you keep it for say 8 to 10 hours. So during that period, yeast will actually get activated, and it will convert the sugar to carbon dioxide, and the carbon dioxide will try to escape from the dough, and it will create the fluffiness of the dough. Now this is about your active dry yeast. And now nutritional dry yeast is actually done in such a way that you can get most of the nutrient present in the yeast, which can be used as a food supplement or can be used as food for the fodder. So here the process is very similar. Only thing is you have to make sure that it doesn't contain any other pathogenic organism. So that's why the first step is to do pasteurization, and after pasteurization, you would just follow dryer and grinder. So moisture content is much lower here, and you can just use it as a food supplement. So all this type of yeast or use of yeast is actually dependent on the requirement. So if you need yeast cake, then you can go for the compressed yeast. If you need dried, active dried yeast, then or maybe which we use for bread making or bakery industries, there you can use the active dry yeast. And when you try to use the yeast for making different flavoring agent or use it for yeah, food for the animal, then you can actually use this nutritional dry yeast. Now, when you use this kind of yeast for different end use, you actually select different kind of strains. So when you talk about use yeast, which will be used in brewery industries, there mostly we use Saccharomyces cerevisiae or Saccharomyces uvarum. So one is sorry. So you can have just a second. So one of the common strain generally used for used in beer industries. So as I told you, depending on the end use, you can use different kind of strain. So the process of manufacture, process of generating substantial amount of biomass for further bioprocesses will be similar. So first you have multiple stages for inoculum preparation, then you have the final fermenter, and then you use all this downstream operation to get different types of final form of yeast. But based on your use, you choose the strain. So if you want to use this brewery industries or brewing yeast, they are mostly we use Saccharomyces cerevisiae or Saccharomyces ruverum. Sorry, it is RUM, ruverum. I don't know. So Saccharomyces ruverum is also known as Saccharomyces cusbergensis. So these two type of yeast we generally use for used in mostly in bearing industry. And in beer industry, depending upon your requirement, you can actually use different kind of substrate. So when you prepare, say, beer, there you use a different substrate. When you prepare wine, there you use a different kind of substrate. So when you try to grow the yeast as inoculum, there also you used similar kind of substrate. Suppose I need to use, I need to brew beer. Now, in the beer manufacturing process, suppose you are using Saccharomyces cerevisiae as your target organism or preferred organism. So, while preparing the inoculum in this process, suppose you are using um, molasses. Molasses means, uh, say, sugar cane molasses. Now, in this process, when you use this cane molasses as your carbon source, while growing the yeast as inoculum, that time also you use same cane molasses in different smaller successive fermenter, and then finally you get your larger fermenter where you actually use this cane molasses to get your desired product. Now, if you go for wine manufacturing process, most of the cases generally the wine manufacturing is actually done by using grape juice. So same thing when you try to prepare the inoculum, there also you try to grow the inoculum using grape juice, and then after getting sufficient amount of inoculum, you just transfer it to the larger reactor where you use grape juice as your carbon source instead of using cane molasses. And it is mostly done to avoid any substrate shock, because if you grow the yeast in a different substrate and put the yeast in a fermenter, large fermenter where we'll be using completely different substrate, then yeast will experience a different kind of substrate shock, and it is not desired. So to avoid yeast to experience any substrate shock, you use same substrate in the preparation reactor or in the inoculum preparation fermenters, and also the same substrate is used in the final production fermenter. And mostly in brewery industry, you need to use little bit higher concentration of yeast just to speed up the process. So mostly in brewery industry, you say in earlier case we mentioned for yeast propagation or yeast biomass generation, in final fermenter you try to get 6 million cell per ml. Whereas in brewery industry, you try to get 10 to 14 million cells per ml in brewery industry. So obviously, when you try to grow the inoculum, you have to make sure that you are getting sufficient amount of inoculum to get yeast concentration in the production reactor at the level of 10 to 14 million cells per ml. And also, when you try to use, suppose you do one fermentation here in one batch. Now, after completion of the fermentation, you can reuse the yeast. So only thing is, when you try to reuse it, you will have certain recycling um, number. Like if you can recycle it only for say 50 to 20 times, then you have to again add fresh inoculum to maintain the concentration in the reactor. So you can reuse the yeast up to say 20 recycles, and then you have to just means you have to use fresh yeast to get desired concentration in the reactor. And after using them for 20 cycles, you can actually again have some end use of this yeast, because this yeast will not be used any further for this beer industry. This will not have any use in the beer industry, but you can still use it for other purposes, like you can just harvest the biomass and then you can extract different nutritional components from the biomass and use it for other purposes. So you are not discarding the yeast, even if you use it after 20 cycles, after if you use it for 20 recycling, still you can actually get some valuable product from the yeast material you get from the fermenter. Now in distillery, like if you use wine manufacturing process, there your target is to get higher concentration of, because beer will have low concentration of ethanol. So the kind of yeast we'll be using, even if they don't have sufficient amount of tolerance to ethanol, still you can grow them and get your beer. But when you talk about wine manufacturing process, there actually you have to use a either genetically modified yeast or a high alcohol tolerant yeast species, which will grow on grape juice. Here we are using say molasses, whereas wine industry you will be using grape juice, and there you need to have same Saccharomyces cerevisiae could be used there. Only difference is this Saccharomyces cerevisiae strain will have little bit higher tolerance towards ethanol. So that is one particular thing. Another thing is whenever you try to grow them in for wine manufacturing process, depending on strain, you can actually get different kind of taste in the wine. So one common strain is Saccharomyces cerevisiae, and then also you can have few other strain for making different types of wine. Like one of the wine is known as table wine. So when we'll discuss about wine manufacturing process, there actually we'll discuss more in detail about different types of wine. So table wine manufacture means for preparation of table wine, generally we use Saccharomyces bioanus. So that is one type of yeast. Similarly, only thing is whenever you change the strain, it will require different kind of nutrients. So nutrient supplements will be different, and sometimes they will require a different kind of substrate also. Like sparkling wine, their common strain is again Saccharomyces, and that's called Beticus. So different type of wine will require different type of strain, and obviously they will require different substrate or food additives or nutritional additives. Now, one special type of yeast is again very specific in Japan, because in Japan they used a different kind of yeast to produce sake. So that sake yeast is really ridiculous. Here again, the initial substrate will be different. So in most of the cases, when you talk about wine making, they mostly use grape juice. So, and kind of grapes are also different. Like you can have green, you can have red grapes. Then you will get different kind of color also in wine, red wine, white wine. So here mostly we use grapes, and in For sake, you use steamed rice, and then you do the fermentation to get your desired drink. So for different kind of uh, drink preparation.
Can't you just use? So I had a question here. Hello.
Yes, please go ahead.
So is it necessary to steam the rice before fermenting it, or can you just use the grain?
No, because when you try to use the grain, you will get different ingredients. So when we'll discuss about specific alcohol preparation, then you'll see the importance of different stages of grain preparation. Like if you, as you asked, I'll just give you a little bit of overview. So in some industry, they use barley. Barley is also one of the common grain we use for ethanol fermentation. So when you uh, grow the barley, or when you try to provide the moisture to the barley, they will germinate, and they will also during germination, certain proteinaceous compound will be actually there in the shoots. Now, before using them for fermentation, you roast them. So obviously the proteinaceous component will also get roasted, and then with you add certain amount of water and then smash them, and then do hydrolysis, get the monomers out from the system, and then use the strain to get ethanol. Now, if you use only rice grain without doing any preparation, the content will have different kind of proteinaceous layer. Because if you can remember, in India also we say if you just drain the water after boiling rice, you will be losing a lot of vitamins and other nutritional component. So if you just use normal rice, then you try to ferment them using the yeast. The constituents in the substrate will automatically get changed. And major problem with all kind of drink is most of the user, they prefer to get a specific taste and specific flavor also. So whenever you have any change in the preparation, the ultimate taste as well as flavor will also get changed, and then people will not appreciate. So whenever we'll prepare a specific type of drink, we need to follow exact same procedure to avoid having any change in the flavor as well as the taste. So if you use only rice, yes, you can actually, when you raise the temperature a little bit, because obviously during fermentation also temperature will raise to 45, 50 degrees centigrade. So at that temperature, it will get cooked a little bit, and also the hydrolysis will be a little bit easier. So when you talk about fermentation, first step is hydrolysis to get monomers from these carbohydrates. Now, if you use steamed rice, the hydrolysis step will be much more easier here. But if you use raw rice, it will get hydrolyzed. It will take a little longer time, but taste of the final drink will also get changed, because a lot of other component present in the raw rice will also get introduced while making the final wine. So the sake you will get if you use steamed rice will have a different kind of flavor and taste. And if you use normal non-cooked rice and then do the fermentation in the reactor itself, then you will get a different flavor and taste. And also another advantage with this steamed rice is whenever you are using rice, there is a possibility rice grains will also have some fungi or bacterial strain present in the system. But when you do this preparation, when you prepare the rice first, obviously you are eliminating having the all other contaminant present in the rice, because when you just use steam rice, you have already killed all the other all other organism present in the raw rice. So essentially, if you put the steamed rice in the fermenter, you are quite sure that you are not adding any other contaminant in the system.
Right, sir. So is grape juice also processed similarly
Yes, yes, yes,
before?
yes, yes. So we are not going to add the raw grape juice, but just I'm not giving too much of details here because here we are just talking about different type of yeast and their usage. But when we'll discuss about wine manufacturing or beer manufacturing processes, there actually I will tell you what kind of precaution we take and what kind of pre-process we do before adding any substrate to the system. There is a means if you talk about this wine manufacturing process, the upstream process is quite tedious. Means first you have to wash the grape, and while washing, you have to use certain disinfectant. Then you have to crush the grape, and then after smashing the grape, you will get mixture of grape juice and the pulp also. Then how to separate them? What kind of fermentation you do when you get white wine, when you get red wine? What kind of process you do? All these details we'll discuss when we will actually discuss about wine manufacturing process, but it is actually not very straightforward. You cannot directly add grape juice and start the fermentation process. And if you see in different vineyards, whenever they grow the grapes, a lot of other insects and other bacteria and organism will get attached to the grapes. So after grape harvesting in different European countries, they use different traditional as well as sophisticated advanced methods. So we'll discuss about that also, because you might have seen different movies. People have just put the grapes in a big container, and many people they just smash them using their own leg, and then finally they extract the juice and put it in the fermenter. That kind of operation is still going on in traditional winemaking process, but in most advanced wineries, they use a really very sophisticated technique to get rid of all types of contaminant. And everywhere, the tendency means this kind of procedures are actually followed to get a consistent taste and flavor in the final product. So different wineries or different wine manufacturing processes, they don't try to deviate from their unit operation just to ensure that the final product should not have much change from the previous batches. And that's why in different brands, they actually claim a distinct different taste because they have some unique processes involved in the manufacturing process, and sometimes they don't disclose those secrets also. So while we'll discuss about wine manufacturing, we'll actually have a detailed discussion on that. So this is all about your yeast, usage of yeast in uh, distilleries or beer industries. And apart from that, if you want to use inactive yeast or dried yeast, then you can just use it and as food supplement, as I mentioned earlier. So you can just dry the yeast and don't care about the moisture much, because there your main objective is to get substantial amount of nitrogen. So these are the use of active yeast. And then another type is called inactive yeast. So whatever I discussed till now, that is actually use of active yeast, because you are just using the yeast cell to do another process. And the second type of use is inactive yeast. So here your major intention is to get nitrogen. So you try to get as much as nitrogen is possible from the system, different vitamins, and sometimes trace elements. So mostly, if you need to use it for human, means if you want to use it as a food supplement for human being, then you will take proper precaution that it should not contain any other pathogenic substance. For animal fodder, that purification steps are not that critical. So when you use as an animal fodder, then you try to maintain sufficient amount of nitrogen, particularly vitamin B complex in the system, biotin in the system, and then you, as it is inactive waste, you make sure moisture content in this particular system is very minimal. Otherwise, other contaminant will start growing on them, like different fungi or bacteria will try to grow on them. And particularly when you use them for human as food supplement to any food supplement for humans, there you have to make sure that these are free of uh, other pathogenic strain like salmonella. That is also a common contaminant for this kind of yeast. So you have to make sure that salmonella is not present in the system. And mostly because of high protein content, this nitrogen is your target element that you get substantial amount of nitrogen. So in a few cases, they also put certain condition that it should have at least certain percentage of nitrogen, like 8% nitrogen or more. Nitrogen content should be greater than 8%. Then only they will consider as a good in a supplement. And for this food supplement, again, initial yeast material can be different. So you can have Saccharomyces cerevisiae, but most common yeast for making different food supplement is Candida utilis. Candida utilis is one of the most common yeast used for making different food supplements. Now, uh, again, when you try to grow this kind of yeast, you can have different kind of substrate. And when you change the substrate, this nutritional concentration or different nutrient present in the yeast extract, their concentration will also get changed. So one option is to use molasses. Another is wood sugar. Why did not open? So you can have molasses, you can have wood sugar, you can have ethanol. So different kind of carbon source you can use. You can have methanol. You can have hydrocarbon grown. So different kind of organic carbon source you can use to produce this dried um, inactive yeast extract or inactive yeast, and then you use the yeast material to extract or to get advantage of different constituents. Now, whenever you change the substrate, you may need to change the biomass also, because different biomass will have different substrate affinity. So mostly for this inactive yeast, you choose Candida. So either Candida utilis or Candida bovidini, different kind of strains are there, but mostly Candida is one of the common strain we use for Candida species are used for going this yeast and get different final yeast extract. And particularly hydrocarbon, wherever you need to use, if you need to get a little bit of more lipid content, or if you need to utilize certain waste stream having paraffins or other higher organic carbons, there actually you can use some specific yeast. Like I can give you one or two examples. One is Candida. Mostly there are Candida maltosa. Really. Or you can have lipolytica, because again it is again to have lipolytica. So depending on the carbon source, you can use different kind of candidate species to get final yeast, and then you extract the yeast to get say single cell protein also. So mostly for single cell protein production, hydrocarbon is a common substrate used, and there you can just use this kind of candida species, and the single cell protein will actually be used as animal food. So these are all different kind of use of yeast. Now, when you try to use yeast extract, from that yeast extract you can actually get different enzyme also. Suppose if you want to produce invertase, so invertase, lactase, these are all common enzyme you can get from yeast extract. So one option is to get invertase, and you can get lactase from this yeast. And sometimes if in some specific pharmaceutical use, you also require some ribonucleic acid. So you can get ribonucleic acid or RNA from this candida species. So some strain like Candida utilis is actually have a speciality. It has 10 to 15% RNA and very low DNA. So you can actually extract ribonucleic acid from Candida utilis. So these are all the different use of yeast. So one use is actually direct use, that is active yeast. Second is inactive yeast, where actually you can get different nutritional component from the yeast. Third is production of different enzyme from the yeast. So you can get different intercellular enzyme from the yeast. And this direct use of yeast is very common in bakery industry and brewer industry, whereas for animal fodder or for food supplement, you can grow the yeast using specific substrate. So this is all about yeast production. If you have any question, I'll answer them. And in the next class, we'll discuss about little bit about ethanol production. And after minor exam, we'll have detailed discussion on ethanol production from different food crops and also production of wine and beer.
Yes.
Okay, so anybody has any question on this? So whatever I discussed earlier, that you will be submitting an assignment mentioning the type of yeast depending upon their final usage. So whatever we have discussed today, you can just write few points on different type of usage of yeast. So active yeast, inactive yeast, and also within inactive yeast, what kind of yeast you can get when you use different carbon sources, and also the yeast hydrolyze like different enzyme production from yeast. So you can just give little bit of details. What kind of strain should be used for production of certain things? Like as I mentioned, for RNA, we use Candida nuclease. So if you try to get a lot of invertase or lactase, then what type of yeast you will be using? So you can just have 1.5 page long assignment where you will just use production of yeast for different final usage. So what are the usage you get when you produce yeast, and what type of strain generally use? So whenever you try to write this, suppose you are talking about um, uh, methanol or dry yeast produced on methanol, then what type of strain generally we use for the production of dry yeast using methanol, and say hydrocarbon, then what type of yeast we use to grow them and their final use. So it will not be more than 1.5 page long.
Yes, sir.
Yeah.
Sir, when is the deadline for the assignment?
You can submit. See, your last exam will be on, I think, Monday, Tuesday, Wednesday, Thursday. Last exam will be on Thursday.
Yes, sir.
So you can get 3 more days, like Friday, Saturday, Sunday. So the deadline will be, so you will not have much pressure. So you can immediately after finishing the exam, you can start working on that, and then you can submit it by Sunday evening. So I'll just share a Google form, and I also mentioned about the deadline. So by Sunday midnight, you need to submit the assignment. It will be handwritten assignment, so you have to just write on a plain paper and then uh, take a photograph of that and convert it to PDF and then submit the PDF on that.
Yes, sir. Sir, can you also
So we
send
can't
like,
write down.
Sorry.
can you send a mail regarding the instructions for this assignment?
Sure, sure, sure.
Okay, thank you, sir. And sir, about the minor?
Whatever we'll cover till this Friday will be the syllabus for the minor exam.
Okay, sir. Okay, sir.
And for
So, can we
assignment,
not type this assignment out?
no. So because the tendency is whenever you ask for typed assignment, same content will actually just circulate it among student and they will submit the same thing on multiple times. So even if you try to copy from others, at least you will spend some time to write it.
Yes, sir. Sir, and the minor will be Moodle based, or what will be the format for the minor?
Um, Minor mostly I'll see, means maybe we'll have fill in the blank type of questions,
Okay, sir.
and either Moodle or Google. We'll see that, because last time when we tried with Moodle, it got crashed. So Moodle has a problem whenever you get more than 30 students.
Forms would be better. Yes, sir.
Google Form is more reliable in that way.
Yes,
So
sir. Yes.
I'll see that, because my experience with 30 or more student is little bit tricky with Moodle. But Google Form is more reliable.
Yes, sir.
So I'll send the instruction before the exam, because I think J slot exams are there on Thursday. That's the last day actually.
Yes, sir.
So definitely I'll share the information related to the minor exam. But for this assignment, I'll send a mail for the link, but I'm not going to discuss much on the assignment there. So those who are attending classes, they know exactly what to do.
Yes, sir.
And rest of the students, if they're not attending, I don't care.
Oh, okay, sir. Okay, sir.
Yes.
Thanks, sir.
Okay. Thank you.
Good afternoon.
So, just confirming for the assignment, we just have to basically have a 1.5 page summary on what different strains of yeast are used in different productions.
Yeah, miss, if I just ask you to submit an assignment to mention about use of yeast cell, use of yeast cell. So what
Fine,
kind?
fine, sir.
So it will not actually require 1.5 page. And I think you can do it in 1 page also, like activist, inactivist, and also different kind of waste to grow with different substrate. So key point for all discussion would be the species you are going to mention, and also their use. And when you talk about enzyme also, then what kind of species you use and what specific constituents you get if you use that kind of yeast. Like I mentioned, when you use inactive but dry yeast, your main target is to get nutritional components like nitrogen and other trace nutrients. So those things you have to mention there. Only thing is, if you do it before minor exam, it may help you to prepare for the minor exam also. I'm not insisting you, but it totally depends on you. If you want to do it before minor exam, you may get a little bit of help from there, because in the question paper also I may give 1 or 2 questions on this.
Yes, sir.
Okay then. So see you on Thursday, Friday then again.
Yes.
Thank you, sir.
Thank you. Thank you.
Thank you.
Thank you. Thank you.